In this video, we will learn how to run a quick search to get relevant and comprehensive results quickly. We will explore and filter a list of scientific documents and see how to export results, save a query, and set an alert to be notified about new relevant documents so that you do not miss any critical information. Quick search is the easiest way to start semantically discovering documents that are relevant to your research. You land on this page by default after entering your username and password. Alternatively, you can always access it through the document finder in the menu bar on the top of the screen. To discover relevant documents, you just need to start typing your terms of interest here in the search bar. Let's say you're looking for scientific documents related to a gene that you consider a potential drug target. As you type your term, the system starts suggesting concepts that match your search. When you see the match here, just select it and the system will automatically populate it in the search box. Like in my case, this SORL1 protein has filled the search field. Not all the documents which mention your concept somewhere in the title, abstract, or full text may be relevant to you, so you can add more context to narrow the search down. For example, you may only want to see documents where this gene is discussed in the context of a specific disease, such as Alzheimer's disease in my case. For this, you simply add an additional term or concept to your search. Alternatively, you can add any of these concepts through the Domain Explorer instead of typing them in the search box. The results are the same, but in the case of the Domain Explorer, you browse the whole ontological tree of our 40 million concepts, which, as you can see here, are organized into 23 domains. Here, you can also search for a specific concept and see where it is located in the tree, or use this tree to discover relevant terms such as genes and proteins, etc. When you click on any of these concepts, the system provides concept details that you see on the right and allows you to select the concept for the search. By default, the system combines the search terms with the Boolean operator AND, but you can change it to the Boolean operator OR. If you want to use the Boolean operator NOT, you need to send this query to Advanced Search, as Advanced Search supports this Boolean operator among others. Advanced Search can also be used to build very complex and very specific search queries. If you want to filter scientific documents, you can use dedicated filters by clicking on Show Filters. First of all, you can apply general filters for all scientific documents. For example, if you are looking just for those documents that are quite recent, let's say published from the start of this year, you can specify that. You can also ask the system to retrieve only those documents where your concepts are discovered in close textual proximity. If you want to increase the specificity of your search and only see those documents where your gene and your disease, for example, are mentioned next to each other in the text. Secondly, you can apply specific filters for patents, clinical trials, grants, and publications. You may be looking for patents just from a specific patent office or of a specific assignee or inventor. Or you're looking just into ongoing clinical trials sponsored by a specific company or organization. Or you're looking just for those publications that are coming from a specific publisher or are open access. After you specify your search criteria, you hit Search, and the system generates a list of scientific documents grouped into publications, patents, and grants. And you can jump between them simply by clicking on the corresponding content set. In this case, for my search criteria, there are no clinical trials and no drug labels, which is expected because the gene that I am looking at is not a clinical target yet. By default, your documents are sorted by relevance, which basically reflects the frequency of occurrence of the concepts that you specified. But you see that you can change how the documents are sorted. Of course, sorting varies from content set to content set. For example, patents will have different features for sorting when compared with publications. You can explore the results in more detail. Clicking on the document name takes you to a page providing a detailed overview of the document. As you can see here, a publication abstract highlights your concepts of interest, and you can find more details such as references, links to the publisher's website, and more. If you do not want to open a detailed page, you can get quick info directly from the results list instead. Clicking on Quick Info provides an abstract and allows you to rank or bookmark this source. Add it to your collection if you want to read and further analyze it later, or share it with your colleagues. And again, you can always navigate to the more detailed page as I did before if needed. Of course, you can export these results outside of the platform, and Dimensions Life Sciences and Chemistry supports 2 export options. You can export metadata into an Excel file, which basically provides information on a title, authors, assignees, publication dates, etc. You can also export semantic concepts from the full texts of selected documents so that you can perform an analysis on, for example, drugs that are mentioned in full texts of these relevant documents, or proteins and genes or companies, etc. Finally, you can always save your query or set an alert to be notified whenever a new document of your relevance appears in the platform, so that you do not miss any critical information for your decisions. We have just seen how to use Quick Search with auto-suggested semantic concepts to quickly get relevant and comprehensive results. We have also learned how to combine multiple concepts for the search and apply diverse filters to narrow down the list of documents to the most relevant ones. Finally, we have seen that a document list can be exported in several ways outside of the platform for further analysis, and that you can receive notifications for when new relevant documents appear in Dimensions L&C. To get the most out of the platform, please watch additional videos dedicated to specific features and analytics.